Hello, and welcome back to Introduction to Genetics and Evolution. In the previous videos, we looked at how to study the process of recombination, and specifically how we can leverage recombination to actually map genes associated with diseases. And again, as I've said several times, we're looking for an association between alleles at markers, such as these HapMap SNP markers that we discussed, and a phenotype such as disease status. That is essentially what we're trying to do with genetic mapping. Now, what we're going to do in this video is we're going to actually try to map simple genetic traits, so traits controlled by one gene that have a very large effect on that trait relative to genetic markers in a cross. So we can actually use HapMap markers, just like we used the Drosophila mutations in previous videos, to map simple genetic disease. So you remember we did these problems using these three genetic markers, A, B, and C. And we did the case of the test cross with an individual that was heterozygous for all three, so big A, little a, big B, big C, little c, crossed to somebody who is homozygous, little a, little a, little b, little c, little c. Now we can imagine that A and B are SNP markers with, so they necessarily have two or more alleles, and that they have a known location in the genome. Now what we can say now is maybe C is the disease gene. So what we're trying to actually do with a lot of genetic mapping, this isn't all of mapping, but part of mapping, is to determine whether C, the disease gene, is between a certain pair of markers, and secondarily, about how far is it? Like, is it, for example, between A and B? And if so, is it closer to A, is it closer to B, or is it off to one side? Is it maybe out past A and further away from B? That is genetic mapping. So let me illustrate this with an example, and we'll be using concepts very much like what we did with the Drosophila mapping of genes. Let's use sickle cell anemia. Now this disease is recessive, meaning you have to have two copies of the mutant type to exhibit the disease. Alternatively, if you only have one copy, if you're heterozygous, we refer to you as a carrier. That means that you probably had a parent who had the disease, for example. Not necessarily, but you probably did. Now let's imagine you're surveying a lot of families, and you have multiple cases of carriers, these individuals that we know are heterozygous for the disease, having kids with affected individuals. When I say affected individuals, I mean ones who actually have the disease. So you're crossing somebody heterozygous for the disease, the carrier, with somebody homozygous for the disease. So this is, again, just like the test cross we did before. Now let's say you have the genotypes for the parents and for the offspring. Now you don't know what the gene is causing the disease. This is a very important point. You do not know what or where the gene is causing disease. When I say you have the genotypes for the parents and offspring, I mean there are genotypes for a bunch of genetic markers. Now you don't know where among those markers the disease-causing gene is. So let's focus on two genetic markers on chromosome 11. Let's say that you've already, from something else, you already have some idea that this disease is caused by mutation on chromosome 11, which actually is true, by the way, for sickle cell anemia. Let's say you have two markers. Let's say the A marker is at this point, there's a picture of chromosome 11's chromosome. Let's say the A marker is at this location. I just made this location up, don't worry about that. Let's say big A might be an individual that's homozygous for the letter C, for the nucleotide C at that position. Big A, little a has T at one copy and C at one copy. And little a has T at both copies. This is irrelevant. I just wanted to give you some idea of what I mean when I'm saying these SNP markers in big A and little a. So this is something just to make it more real in the context of a DNA sequence. The important thing is that A is over here on the chromosome, and B, in contrast, is a little bit lower down on the chromosome. So what are we trying to do? We're trying to see, is the gene causing sickle cell anemia between A and B? Is it down below B, or is it at that little area above A? That's what we're trying to figure out. So let's say that you have some data now from the sets of families I mentioned. And this is what the family data looks like. Now again, we have the genotypes just for A and B. We don't know anything about C except by virtue of disease status. Okay? So individuals that are big A, big B, little b. So they're heterozygous for both the A and B markers. 416 of them were healthy and one was diseased. Okay? So most of them tend to be healthy if you're heterozygous for both. Those are little a, little b. Most of them, or actually all of them in this case, are diseased. If you're big A, little b, so you're homozygous for a little b, but you're heterozygous for the A marker, you're mostly healthy. There's a couple of diseased individuals. And in this case, little a, big B, little b, most of them are diseased. So look at this for just a minute and speculate whether you think marker A is closer to the disease-causing mutation or marker B is. Now again, remember, mapping is looking for an association between genotype and phenotype. Okay? So what you want to do in looking at this right now, this is just like the problem we did in a previous video. Look and see, does your genotype just at A predict the disease? Does your genotype just at B predict the disease? All right, well, let's take a look now. So at A, just looking at A, or actually let's start with B. Let's start with B. Just at B, big B, little b. So this one's big B, little b. Almost all of them are healthy. This one down here is also big B, little b. Almost all of them are diseased. This one's little b, almost all of them are diseased. This one's little b, and almost all of them are healthy. So B really doesn't predict the genotype very well at all. What about A? Well, this one's big A, and almost all healthy. This one's also big A, and almost all healthy. This one's little a, and all diseased. This one's little a, and almost all diseased. So clearly in this case, the genotype at the A marker is more predictive of whether you have the disease than the genotype at the B marker. So we can assume that A is probably closer to this disease-causing mutation. And we haven't said whether it's between A and B or not. It could be in that little area above A, as I said before. Well, what we can do, and this is an assumption, if this is a simple genetic trait, meaning a trait that is fully explained by a single gene having a very large effect, what we can do is we can split this section up, this whole section over here, and just infer what's going on at the disease-causing mutation. So we're saying that these ones are healthy and this one is diseased. Well, if you only get the disease when you have the mutation, then this one must necessarily be little c, and this one over here must necessarily be big C, right? Because these ones are carriers, they're healthy, these ones are diseased. So let's break that up. Again, we have another gene causing disease, so we're just splitting this row up. So we're taking this original problem where we're looking at 2 markers and basically now looking at 3. The third being what we're inferring to be happening at the disease-causing gene. I split this up relative to disease status, and that was it. So what we can do is we can split up all these other ones, like this one here, right here. These ones necessarily are big C, there aren't any. These ones would be little c, and we can continue the same process that I just did. So I just broke up everything here from the previous slide into this set of individuals. And again, we're assuming all big C, are healthy. They are carriers of the disease, but they do not actually exhibit the disease. And little c in this case are all healthy. Okay? So, without doing any math at all, I want you to tell me the order of the genes here. Is the C, the disease-causing mutation, between A and B? Is it off past B, or is it off past A? It's very simple. What you do in this case is you just try to figure out which one is the double recombinant. And from that, and since you know the phase, you can identify which gene is in the middle. Double recombinants are always the rarest class. In this case, the rarest class would be these right here. Now when we look at this, which 2 of these genes are in the same phase, and what's the third one that's in a different phase relative to the parent? So here are the parentals, and we can confirm that is actually the parental phase because that's most common. When we look over here, the rarest 2 have C being coming from the other chromosome, whereas the capitals are together, just as they should be. So little a and little b, big A and big B, C is the other one. So C is necessarily the double recombinant, so we must have had something like this to get the double recombination event. So the order must be B. A, C, B. So C is, in fact, in the middle. Now, what we could do from those numbers you saw, and you can do this for practice if you want, you can go back and actually calculate the recombination fractions. The recombination fraction between A and C is only 0.8 centimorgans, or if you calculate it directly, it's 0.008. Between C and B, it's 15.1% recombination, or 15.1 centimorgans. So we can infer from this, since A was at 11p 15.6, B is at 11p 12.3, C is probably at or near 11p 15.5. In fact, this gene is actually well known to be at 11p 15.5. It actually has already been mapped and identified. Now, let me conclude this video by just recapping one very important point. The goal of genetic mapping is to localize alleles of genes that cause disease. What you're trying to do is you're trying to find where is that mutant allele, like which gene has it. Because again, the genes don't come labeled. Even when you have the full human genome sequence, the genes do not come labeled. Some markers will be associated with disease, and those ones that show that strong association, where alleles of those markers, the genotype of those markers, will be associated with whether or not you have disease, or will predict whether or not you have disease. Those are ones that will be near the disease-causing gene. Okay? Some markers will not be associated, or will be very weakly associated with it, and those are the ones that are likely far away from the disease-causing gene. I hope that makes sense, and we'll follow up on this next time with looking at mapping through studies of population data. I hope you'll join us.